Thank you all for being here. As you know, this is the molecular machines track. Our format is going to be 15-minute talk and then a 15-minute Q&A. Everyone is encouraged to chip in. Very informal, ask any question. Starting, we have Seth Shipman from the Gladstone Institute. This is his first time joining Foresight for an event, so we're very excited. He's going to be presenting on building molecular machines for biology and medicine.
Thank you so much.
Take it away.
Thank you. Yeah, so I'm going to talk, we can intermix the questions in the talk. It's fine with me. So if you want to shout it out, I can see you all. I'm happy to be here, happy to sort of kick off this session. And I thought I'd do so with a prediction, which is that we're at the beginning of a technological revolution in the way that we understand science and medicine. And what we have gained in the past really has been, lots and lots of information using technologies that are built primarily from non-biological parts, right? So things like microscopes, stitches, stents, even sequencers and mass specs and things like that, that are these, machines and man-made parts that can interface with biology only superficially. And I'm going to argue that, the next era, let's see if I can go over here. I'll do it here. The next era, maybe we can go here. Oh yeah, there we go. We got it. It's going to be built on technologies that are composed entirely from biological parts, right? So engineered cells like CAR-T therapeutics, molecules, proteins and RNA, like gene editors and RNA therapeutics that can really interact directly with biology and work in cooperation with these biological systems to gain information and do work for us in more intrinsic ways. And so I think that's what's coming. I think it's going to change the way we think about medicine, from individual disease all the way up to how we monitor and mitigate sort of large-scale existential threats to our ecosystem. So I run a lab at the Gladstone Institute, UCSF, where we try to accelerate this sort of future of using biological technologies. We're in medical school, so we're mostly interested in technologies that interact with human disease. We are, though, technologists, and so we don't study a single disease or even a single system. Rather, we really look for places where there's some sort of a fundamental technical barrier that's holding us back from either understanding disease or treating disease that is imposed by the use of these legacy non-biological technologies. And then we do our best to engineer new technologies built entirely of biological parts that try to get around these barriers. There's a huge world of biological parts out there, and so we try to limit our scope a bit by building these technologies from a toolbox of parts we steal from bacteria, and more specifically from bacterial immune systems, where we can leverage the sort of diversity.
Just a procedural question.
Yeah.
Are these slides going to be available to us kind of afterwards? So
the recordings will be available to the public if approved by the speaker.
Sure.
So you'll find out after.
So I don't have to write down everything.
Don't write it all down, please.
Awesome.
Yeah. So we steal stuff from bacterial immune systems where we can sort of leverage this billion-year-old war between bacteria and bacteria viruses that have given us tons and tons and tons of parts. So what I'm going to do today is just sort of introduce you to one of the technologies that we've been working on as an example, which is a recording device built entirely from biological parts that we are using to spy on the inner workings of cells. The reason why we do this is because we're interested in biology and really complex biology. So one of the most complex things we think about is the acquisition of cell fate in developing mammalian systems. It's an unbelievably complex process, right? So all of us start from a pool of a few cells that have the potential to give rise to every single diverse cell in our body. This plays out over the course of our growth in utero and for about a year of our life. Lots and lots of things happen. We'd love to understand this process because we think it's critical to understand developmental disease. We also think that understanding it will give us better tools to engineer cells as replacement therapeutics, for degenerating conditions. It's really hard to study. And it's hard to study because most of our sort of legacy technologies require us to destroy the system to get information out, right? So we have to destroy a developing system to look at it, to pull out biological molecules like RNA and proteins, And what that means is that we can only ever look at a snapshot in time at any one moment. That's a problem because this is a process, right? This plays out over and over again in when we study biology. And so we think of this as really one fundamental technical barrier that is imposed by the technologies we're using. We'd like to eliminate it or at least reduce it. And the way that we think about doing that is by building something that will allow us to collect data from inside biological cellular systems without having to extract it, right? A molecular data acquisition device that can live and operate inside of a cell. So that's what I'm going to tell you about. In order to build that, we first need some place to store the data, right? So if we're not going to take the data out and digitize it and hold it on a hard drive, we need a hard drive that operates inside the cell. And that one actually was one of the easier problems to solve because cells are full of DNA. And DNA intrinsically is an information-bearing molecule, right? So we think of it as holding all the information that encodes for all of our genes. But we can sort of take one step back and think about it a little bit more abstract and say, well, really, it could represent anything, right? It's a polymer of 4 bases, G's, A's, G's, and C's that can be ordered in any order, and that order of G's, A's, T's, and C's can be used to represent any arbitrary information in the same way that a string of zeros and ones in binary can. So it's an information-bearing molecule. It's actually a really, really good one. We can sort of get a sense of how good it is by just thinking about how much data is actually stored in DNA right now. Right, so if we sort of think about how much DNA there is just in the living world on Earth at this moment in time, and convert that into information, you get this 10 to the 16th zettabytes, which is like a number that is difficult to sort of fathom. To put it in context, right, it's about 10 orders of magnitude larger than all the digital information that we have generated as a human civilization, which is saying something, especially I think relevant in this temple of digital information that we are in right now. So it's a really good data storage device, and we can reuse it, right? And we reuse it because it's compatible with biological life. So how do we write information into it? And specifically, I said I wanted to understand these processes in biology, and so we need to be able to just write information into it over time, right? So some sort of a sort of camcorder or recording device into it. And so we can begin to build this by looking at what's out there in biology. And the first thing we looked at was some proteins from the CRISPR system. I'm going to imagine that most people have heard of the CRISPR system. It gave us Cas9, which is one of the now most promising biological tools for gene editing. We use a slightly different part of the CRISPR system, and so I'm going to give you a tiny primer on CRISPR biology in bacteria. So this gray box is a bacterial cell. It's under attack from a bacterial virus called a phage that has deposited its genome into this cell. This cell is mounting an immune response. That immune response from the CRISPR system starts with these 2 proteins, Cas1 and Cas2. They'll go grab part of that viral genome. They'll insert it into the cell's own genome. So insert the viral DNA into the cell's DNA, and that forms a memory of that virus. So this is a sequence that is an immunological memory of that virus. Right, so it's storing information about some other piece of biology. These integrases are particularly amazing in biological sort of realm because they do a couple of other things. When another virus comes in, they'll grab a piece of that virus and also store it. So they're repetitive. They won't destroy what's already there. They'll just add to it. This place where they're adding it, it's called a CRISPR array. And there's one other thing that's interesting about them, which is that they're directional. So one side of this CRISPR array is called a leader. Every new viral sequence that gets inserted goes in next to that and pushes the other ones down, right? So it's generating a record of viral events organized linearly in the genome over time, right? So it's a recording device for bacterial viruses. We said, maybe it could be a recording device for anything. And so first we just tried to see if we could hijack it. And what we did was we wanted to say, can we put new information into this system? We were inspired by another technologist, Edward Moybridge, who took these first images of some biology, these running horses down on a track in Palo Alto. We made digital versions of them. We then converted these digital versions, these frames, into a code that we synthesized in DNA. So we basically came up with a code that would take the pixel values and represent them in a string of nucleotides and synthetic pieces of DNA. Each frame is represented in a pool of individual sequences, and we would then deliver these sequences to cells that have one of these CRISPR systems. It turns out that they will acquire these just like they would grab a virus, right? So they'll capture these sequences and store them in their genome. Again, we want a time device, and so we know that these things will acquire them over time, and so we can take different frames and deliver them one at a time over days and actually write this movie into cells. We can take those cells weeks later, sequence their genomes, and recover this movie. All right, so we're writing an ordered history of temporal information into the cells. So that's great. We're showing that this is a sort of a recorder that can be hijacked. It's not biological information at this point. We're sort of just putting it in. And so what we've been doing in the last few years in my lab, and this is work that's led by venture graduate students and research associates, especially this guy, Santi Karatline, is try to hook it now into some biology, right? So what actually we want to do is something relatively simple. We want to read gene expression from multiple genes over time, whether they're expressed, and then the order in which they're expressed. This is sort of something that we have not been able to do without typically disrupting the system that we're looking at. And maybe I should say, you know, when I say gene expression or if a gene is turned on, what is that gene, which is in DNA, is converted into RNA. The RNA is converted into protein. Proteins do work, right? So this is a sort of this unidirectional flow of information from DNA to RNA to protein. I will remind you is at the core of sort of central dogma of molecular biology. Everything goes in that direction. In order to record it, we need to slightly subvert that central dogma. RNA is not that stable, and so we want to be able to go from a gene that is on, that is making RNA, convert that RNA back into DNA, make some sort of DNA receipt of that transcriptional event, and then store that receipt in a genomic ledger so that we can figure it out that had happened later on. And then again, we want different barcoded receipts of different genes so we can follow an entire biological process. That's the idea. The way that we do it, so the back end is exactly what I showed you before. We use these proteins from the CRISPR system to log these things in order, but we have to generate them. And so in order to generate these things, we steal another bacterial immune system part. This one's called a retron. Anyone heard of a retron? No? Sweet. Okay, now you have. We'll do our best to try to make them famous in the coming years. They're amazing. They're another bacterial part. And what they are is a DNA generator that bacteria have. They produce these small pieces of DNA inside cells. They first make an RNA from a piece of DNA, and then they have a protein called a reverse transcriptase that will reverse transcribe that to make a short fragment of DNA. And they'll make thousands and thousands of copies of these DNAs inside cells. They use them to send phages, but we're repurposing them here to link into gene expression, right? So we're actually using these retrons to tag genes so that when they're made into RNA, they make a little piece of barcoded DNA alongside it. Yep. Yeah.
Like, what are we actually looking at there?
Like what is it?
What is that shape?
Yeah. So they basically have this is their operon, right? So it's a protein which is a reverse transcriptase. We call this the non-coding RNA. It's it's about 300 bases, and it's structured, right? So this is the representation of the two-dimensional sort of folded structure of the RNA. These retrons have a very characteristic folding structure that is recognized by that protein, and then it partially reverse transcribes it. So it makes a piece of DNA of about 100 bases.
Is that like stick thing? That's the holding structure?
Yeah, exactly.
Okay.
Yeah.
Thank you.
That's right. Yeah, so there's a physical aspect to this. Very small, but physical. All right, so here's the sort of pipeline we're working from. We want to record this green gene. We tag it with some components of the retron. It's in this non-coding RNA. When it's on, it makes this RNA. The reverse transcriptase reverse transcribes it. These integrases log it in the genome. Now we have a genomic record of that transcriptional event. And once we engineer these things to work together, we can turn them all on and sequence the genomes over time. And we see that if they're all on, we accumulate more and more of these sequences in this genomic ledger, right? So we're basically recording the duration and intensity of this signal. That's one signal, but because we're operating in DNA, we can easily diversify it. So we can take these sort of barcodes we're making and modify the middle part of it. So we can have lots of different ones. We can tell them apart once they're acquired into the ledger. And so we can have lots of different barcodes that are all operating the cell at the same time. So that's the idea. Here's what I said I wanted to do: record two different genes over time in one cell. Here's the way we do it. We're in bacteria. We've got two different genes that we can control, right? So we can put cells into different broths that will turn on, we know, one or the other of these genes. We tag one with a barcode we'll call A, another one with a barcode we'll call B, and then we turn them on one at a time. We do this for a day each. So what happens is if we take these cells, we turn on first this A gene, and then we look at these genomic ledgers, we see mostly at that first time point, so we turn it on for a day, we see mostly the A accumulation in the ledgers, and then we flip the broth, we turn on the B gene, and then on the second day, we see mostly acquisition of these B sequences. And again, it operates in both directions. So this is showing that the parts work. It's cheating, right? Because we're taking a time point in the middle, which is exactly what we said we were going to do. And so the key here is, can we go at the end, not take that middle time point and recover what must have happened? And again, the way that we do this is we use those that sort of work of the biology, these CRISPR integrases, which is that they do directional integration. And so we can just look at these genomic ledgers and know what happened earlier and later. So we run the cells through this. We don't take the middle time point. We sequence a bunch of these ledgers at the end, and then we quantify them all. We'll go into exactly the quantification, but it's not nothing super fancy. We're mostly just looking at them. Yeah.
Can you just give us like a story? Like, what would you see? Like, just, like this protein makes that protein, makes that protein. Is that
like what we're trying to record?
Yeah.
Yeah. So it could be anything, right? I mean, we want to use these in developing and differentiating cells where we know that there are genes involved in a biological process, but we don't know the order that it plays out. And these orders you can kind of think of, as being recipes for how to push a cell through a biological process. Right? We want to know not just the ingredients, but the actual order to put them in place, because then we might be able to recapitulate that biological process. Yeah.
So just to make sure I understand them, kind of with a caveat that I don't know much about biology. Basically, you introduce the agent that observes the process in the cell and make the recording by basically piggybacking the process that's natural. So it's like having a small observing program that creates a record of the sequence in which the reaction network happens inside the cell.
You got it, perfect. Yep. Yeah, that's right. So, I mean, this is something that we would try to do. We would assemble by pulling these things out of cells at different points. We just want the entire thing to be happening inside a cell, because that means it can happen inside a complex system without us having to intervene, right? That system could be a mammalian system that's developing. It could be an ecosystem that we don't have access to, like our gut. And so having it be sort of biological lets us put it somewhere where we otherwise couldn't use it.
So it's basically a flight recorder.
That's right. Yeah. Yeah. That's one sort of analogy that has been used actually pretty commonly. So it's the like black box of the cell. Okay, so does it work? That's where we are, right? Can we do it? So we do this, we run it, we analyze all of them, and what I'll show you here is sort of one way of analyzing these. Again, this is a simple one where we're doing just two different genes relative to each other. If we and we'll analyze them, positive values are going to mean that A is earlier than B. Negative values are going to mean that B is earlier than A. We take all the ones where we know that A should have come on before B. We run them through this, and we can determine again after the fact what must have happened. And if we look at the exact opposite sort of arrangement, we know we turned on B and then A. We analyze these whole things. We can determine it after the fact. Right. So this is a simple version of this machine, but we are hoping that we will keep building this and make very, very complex versions where we can run through really complex biology that we otherwise couldn't understand, store the data through the whole process, be able to go in the end and recover what must have happened. And we think that that's going to totally change the way we're able to understand these biological systems. Yeah.
How do you recover?
Sequencing.
Okay.
Yeah.
Same thing.
Yeah. So in the end, we do have to destroy it, right? So at the end, we have to get the data out. Potentially, we could get around that in some other way, but right now, we just pull it out and we can sequence it, and it's captured inside the genome. So, yeah.
So the simplest question probably. So with this new with the barrier of non-destroying system, of course, you also want your measuring system not to interfere with the system.
Yeah.
Is it a problem? Serious?
Yeah, I mean, you never get anything for free, right? So we have to touch aspects of the biological system to record it, right? So right now we are tagging genes with these things, and then we are expressing proteins that wouldn't otherwise be in the cell. There's a cost to that. There's always a cost to that. We try to keep it as minimal as possible, and then at the core of it, you see whether or not the overall biology is maintained, and then if the biology is maintained, you hope that then the cost you impose was not so large that you fundamentally changed it. But always there's a cost.
I had this vision in my head of like a human being with camcorders just popping out of them all over the place because you're a little overexpressed.
Yeah, that's right. I don't know that you would be able to see them, but they would be there. Yeah. So, one thing I think that is important to think about is as we build our technology from our issue right now is inefficiency in the rate of grabbing these barcodes. So our temporal resolution is limited by that rate. We can get over it a little bit by using many cells that are experiencing the same environment, right? So we can take low resolution data that it was acquired by many cells and recompose sort of a consensus temporal history of what those cells might have gone through. But ultimately, we'd like it to be higher resolution so that we can understand the differences between cells, which we at the moment can't do. Yeah.
Any type of error correction using DNA logic?
We're not right now. Certainly one could, like when we were encoding information in DNA and storing in cells, we played around with some error correction. DNA is incredibly dense storage material, so you've got lots of room. Even though these little barcodes we're acquiring in the system we like the best are just 33 bases, that's plenty of room to do a lot of sort of complex logic. We'd like to build more logic in there. So one thing, maybe error correction is not our first one, but we'd like to build logic gates. So sense not just, the raw signal of one and then the other, but, when they were on together, when they weren't on together, these kind of proved things. Yeah.
Is there any bias towards detecting like more highly expressed genes over the lower expressed ones?
Yeah, for sure. You can acquire one signal multiple times. And so something that is on strong and long, you will get multiple signals building up. The hardest, so yes, you basically get a signal that tells you how the amount of gene expression, which is good. One of the hard things is telling the difference between strong and long, right? There's one part of the system that I didn't mention that we leverage, which is another aspect that we just got from the biology, which is that these CRISPR proteins will also integrate random fragments of DNA that are just floating around in the cell at a low rate. We use these like a clock. So we can actually localize also not the elements relative to each other, but relative to sort of this background signal. And that helps a ton. And so we actually can get some dynamics of how strong the signal was at any given point in time, again, with relatively low resolution, but very weak signals we sometimes just miss entirely. Yeah.
Sorry, I lost it for a second.
Yeah.
So you said it's gene taking, does that mean you're modifying the genes of the organism you're monitoring? Is there some possibility that you could inject this into an organism? Inject it into it?
Yeah, that's right. So right now we are genes beforehand that we know with this set of genetic parts that would then record genes that we didn't have to touch ahead of time. We can't do that yet. And so at the moment, what we're thinking about in terms of nearer term applications are sort of sentinel cells, right? Cells that are sent into an environment to report out on what's happening to their neighboring cells yeah.
So right now, to be able to get gene expressions for different things in the correct procedure that you have, you're dumping them into specific chemical soups to be able to express that. So inside of an organism, you've got to flood the entire thing with all of those to be able to actually record data in this way.
Yeah, so the moving between different soups is sort of our way of validating technology, right? We need a known signal to tell whether or not it works,
Right.
But it wouldn't be the goal, right? The goal is to go in and once we know it works, put it into places where we actually don't know the answer, right? Again, we are thinking a lot about these environments where we know a bunch of genes that are involved in a piece of biology, but we don't know the order that they are involved. And so now that we validated it, we can start to think about putting them into those environments to try to get that order.
This is maybe very naive, but would it be possible to use this to record like non-biological processes in the sense of like, if you could figure out a clever way to turn like something else into DNA over the course, like while it's happening?
Yeah.
Like, is there any reason why you couldn't just use this as a sensor there?
Yeah, so my friend of ours, Harris Wang, who's got a lab in Columbia, uses a similar back end. So we were sort of push these forward at different times, and they've hooked it up to a like a redox system where they can set in electrical pulses and record those. And so it's basically an electrical front end and then a biological back end. So anything you can turn into a signal is recordable.
Okay.
Yeah. Well, I'll be around. We can talk more. But
Thank you, Seth.
yeah.
Wonderful.